आज ही डाउटनेट ऐप डाउनलोड करें। डाउटनेट पर होगा अब आपके सभी मैथ्स, केमिस्ट्री, फिजिक्स और बायोलॉजी डाउट्स का सफाया। बस अपने क्वेश्चन की फोटो खींचो, उसे क्रॉप करो और तुरंत वीडियो सॉल्यूशन पाओ। डाउनलोड नाउ।
In this question, we have been given 3 terms and we have to write their functions in biotechnology. Now we all know that the recent advances in biotechnology have enabled us to carry out genetic engineering. Now what do we mean by biotechnology or genetic engineering? Through these processes, we are able to manipulate the genetic material or bring changes to the DNA of living cells so that we can introduce any desirable gene into these living cells, and after incorporating or introducing this gene into our host cells, we can use these living cells to produce any biochemical, whether it is a protein, an enzyme or a hormone, on a mass scale or a commercial level. So there are many tools that are required in such recombinant DNA technology or biotechnology, out of which the first thing they are saying is polymerase chain reaction technique. Now in short, polymerase chain reaction is also called PCR, and this PCR helps us in amplification of our desirable gene or gene of interest. Because if we want to introduce any gene into a host cell, first of all we have to isolate this gene and we have to make many copies of it. So what we are doing in PCR technique is we take our genomic DNA, we take the DNA fragment that we have isolated, the desirable gene, and with the help of some primers and DNA polymerase enzyme, we generate a large number of copies of this gene. So using the nucleotides that have been provided, this DNA polymerase enzyme is going to make many copies of the desirable gene, of the gene of interest, using the genomic DNA as a template. So this is our PCR technique, using which we can amplify or we can make up to 1 billion copies of our gene of interest. So what is the function of PCR in biotechnology? It is used for the amplification of the gene of interest. Next they are saying restriction endonucleases. So restriction endonucleases are basically molecular scissors. In easy language, they are called so. Actually, they are enzymes and they come under the category of nuclease enzymes because they are able to break down or cut the nucleic acid. But we call a nuclease enzyme endonuclease when it can not only cut DNA from the ends, but it can make cuts at specific locations within the DNA strand. So what we do with the help of restriction endonuclease enzyme, we can take any genome that we want, and there are different types of restriction enzymes, all of which are going to have a specific recognition sequence. So wherever they are going to find this recognition sequence, this nucleotide sequence in the length of the DNA, when we expose the DNA to these enzymes, they are going to inspect the entire length of the DNA strand, and whenever they find their recognition sequence at that specific location, they are going to make a cut in the DNA so that we can obtain small fragments, and the particular desirable gene that we are looking for, we can isolate that. So the role of restriction endonuclease enzymes in biotechnology is to make cuts at specific locations within the DNA so that we can obtain the particular strand or the particular gene that we want. Next they are asking about the bacterium thermus aquaticus. So this is a bacterium that is capable of living in very high temperatures, and this bacteria is useful in the polymerase chain reaction itself, because what we are going to do during this PCR, we need DNA polymerase so that using the genome as template, many copies of the gene of interest can be synthesized. But here this reaction is going to take place at a high temperature because we need the strands of DNA to separate from each other. But if we were to take a normal DNA polymerase enzyme, this enzyme would not be able to work at such a high temperature. This enzyme would get denatured. So that is why we obtain this special DNA polymerase enzyme from the thermus aquaticus bacterium, so that the PCR can take place even at a high temperature and the enzyme would not get denatured. So what is the role of this bacterium? We obtain DNA polymerase from this bacterium, which is thermostable, or which is not going to get denatured. It is going to perform its function even at the high temperature at which our PCR is occurring.
क्लास 6 से 12 से लेके नीट IIT JEE मेंस और एडवांस के लेवल तक 10 मिलियन से ज़्यादा स्टूडेंट्स का भरोसा। आज ही डाउनलोड करें डाउटनेट ऐप या व्हाट्सएप कीजिए अपने डाउट्स 8400 400 400 पर।